to finish by 2.
Okay.
But it's upper bound because of movements.
Yes.
So,
okay, I will and try to get through it as well. So what time is it now? It's yes. Okay. Great. Thank you everybody for being here today. It's been a long journey, and all of you guys have helped me along the way. And I try to thank everybody at the end. But if your face isn't on the page, it's not because I don't appreciate it. It's just you're probably in my thesis and not on the presentation. So today I'm going to talk a little bit about the work that I've done in the past years at USC. And I've worked on different types of images, but they're all medical images. And the one theme that really tied everything together was I was really trying to detect disease patterns within medical imaging. And I also try to tune or change the techniques in such a way that they maximize contrast that we're interested in, depending on the disease type that we're trying to investigate. So that's why the little subtitle there. And then once we have those, the technique is tuned to the pathology that we want, then we design some statistical models to analyze the data so that we can find more about each of the different pathologies that I've worked on. So a little bit of an overview of my talk. I'm going to go through some highlights of my previous work that I've done. The first one is bone age assessment. I did some work with hand X-rays. Hand X-rays is the current way of assessing the skeletal age of development of children, and developed the automated system of using those skeletal hand X-rays to determine the development stages of children. The second part I'm going to talk about is a little bit of imaging informatics work that I did on databasing and how medical images are stored in the radiology department. Third, I'm going to go into some work I did in fMRI. fMRI, as some of you may know, is a very powerful technique that we use to probe activity in the brain. And I apply this to a very interesting problem, very pervasive problem. It's interesting you guys are having lunch right now, but I applied it on obesity, specifically pediatric obesity. And then fourthly, I worked on some work with the late Dr. Singh on diffusion MRI. And diffusion MRI is a technique that allows you to image the white matter, and I'll tell you a little bit more about why that's important, especially when it comes to Alzheimer's disease. And then I'm going to finally conclude on two projects I'm especially proud of that I'm currently working on. One is ICA multi-fiber tractography of the cortical spinal fine. I'll talk a little bit about what ICA multi-fiber tractography is. It's an extension of diffusion MRI. The particular exciting part that we have is the ICA multi-fiber part of it, which has been a development program in our lab for quite a number of years. And last but not least, Alzheimer's disease imaging. Also using diffusion MRI in certain parts of it, but in this case I was looking at structural MRI and whether we could use a baseline MRI of a subject to predict how they would, what their neuropsych scores would be in the next 48 months. So, for example, a person has a certain amount of dementia, and then we're able to image them, and we are able to tell how they'll progress in the next 48 months. That will also give us some information about how we can design treatment plans for these individuals. So I'll go right into it. The first project I worked on, a really great warm-up project for image processing and medical imaging, was working on bone age assessment. And I worked on this with Art, who's here today, and he was my mentor for this project. The previous work that was done was looking at the phalanges and the carbo bones, which are indicated there, and that gave us some information about what the bone age of a child is. But we found that these bones tend to develop pretty early on, so in the later age ranges, it really didn't give us enough information to tell how old they were. So what we did was we added an additional bone, and that's what I worked on. We added the radius bone, and I'll tell you a little bit of what I did with the radius bone. So here is a cutout of the area that I showed around the wrist. You see the radius bone on the right. We first did some texture analysis to get the different sort of classes of pixels. We got some are the background, which you can see in black, the soft tissue in gray, and the bone in white. Once we're able to do that, we can then select out the bone parts of the image. Once we have that, we have an atlas of the hand. We all know what the hand looks like. And by that atlas, we're able to locate where the radius bone is. Once we're able to do that, we can locate what we call the growth plate of the radius bone. And this is the region where the bone actually grows. And as you age, basically this area starts fusing once you're fully grown. So there's a magnified view of that. Basically, the growth plate disappears as you get older. So what does this do? Well, here's a little graph. So CAD output is on the y-axis, which is the system output. The chronological age is on the x-axis. And the red line is the 45-degree line. So technically, if the system is working perfectly, these are normal age controls, then all the dots should fall around the 45-degree line. So we also have blue and purple dots. Sorry, they're a little bit small. The blue dots is what my system outputs. The red dots are the ones that was outputted before I added the radius bone. So as you can see here, it didn't do very well at the earlier age ranges. There's a lot of error. But once I added my features, you can see that it's come up to here and it's closer to the 45-degree line. But also we found that we're still lacking from the, you can't see here very clearly, but 17 to 19-year-old age range here. And really this is the last piece of work that we have to do. We still have some accuracy issues here. Although my system did improve it, this is the pink here from the original system, and this is the sort of blue dots here are my system with the radius bone. So it did improve a bit, but we're still a little bit far away from this 45-degree line. So I'm going to switch gears a little bit. I'm going to talk about the medical imaging informatics work that I did. So one of the things that we did in the lab was that we were interested in certain pathologies. And we know that in the PACS system, which is the image database of the radiology department, that there are a lot of cases of different disease types in there, and we could do retrospective studies. But then we realized what we have to do is we'd have to send one of the graduate students, at that time me, to the radiology department, and we'd have to look in this database. And this database is actually very crude. It gives you some information about the patient, when the exam was taken, the type of exam. And all of the pathology information is in this text report, which is sent to the referring physician. But what we found was that actually a lot of these text reports are very highly structured text reports. They're a group of suggested words that physicians are supposed to use so that everybody is clear on what they're trying to describe. And anatomical words are also standardized. And there are databases of these standard words. So what I did was I used that report and I parsed it out right here and created this enlarged database from the original Radiology Information System database, which is the RIS. And now I have structured information. I have information that classifies each of these radiology reports into what location was imaged, what disease types were found, what were the impressions, along with like other medical codes such as CPT-4, which is very useful when you're trying to find images that have the information that you want. So with this system, it allowed us to do retrospective studies a lot easier. I didn't have to go through all the text reports or do text search through the text reports to find the images that I needed. And I also linked this to the PACS system so we can automatically retrieve those images once we found out what kind of codes were associated with the images that we want. Next, I'm going to talk a little bit about fMRI. Once I did that work, I realized that I really wanted to do something a little bit different. I was really interested in some of the advanced statistical methods that was in imaging, and a lot of these were encapsulated in functional imaging. And so one of the big areas at USC, for those of you who know, Michael Gordon has a big group in pediatric obesity, and they study obesity in the Hispanic population. And it's a big problem right now, and it's a very interesting problem. So I really wanted to see if we could use fMRI as a tool to investigate this. So what did we do? we did a very simple fMRI experiment. And this fMRI experiment, you have a subject, in this case, girls that haven't gone through puberty. They're sitting in MRI, and they see a series of images. And the first set of images you see are control images, so doors, cars, bicycles. Next set of images, it shows for 30 seconds. And every 10 seconds, the image changes, and it's randomized. Next, you're going to show an image, a high-calorie image. So, for example, we've chosen some images that are common to these girls that they know, one of which is the hamburger. And again, there are a number of images of these, of different types. One of them is a fried chicken. And we kind of shuffle it around. And next, we show another set of control images. And then we show some low-calorie foods that they know also. For example, salads and fruit and so on and so forth. And we ask them beforehand, think about, while we're showing this presentation paradigm, think about how hungry you are or how much you want to eat that food. And finally, another set of control. And we repeat this about 3 times. And what we're able to do is basically look at the brain activity as each of these visual stimulus is shown to the children. So what did this give us? Well, when we looked at low-calorie foods versus control, we could find certain regions of the brain that correlated with the presentation paradigm. And in fMRI jargon, that's called areas of activation, basically. And here are some of the regions. And we also looked at high-calorie versus control. An interesting thing is the regions, some of them overlap, some of them don't. There's some extra regions that come about when you do high versus control. And the really pretty hard thing that we're trying to do is, are there areas of the brain that activate more in high calorie than low calorie? These are common areas that activate more in high, less in low. Well, this is all good. This is actually, a lot of this has been done. But what we really, really want to do is to look at whether these sort of areas really correspond to clinical measures. For example, fasting insulin. So here in this case, we looked at areas of the brain that have activation that's when compared to the high-calorie foods versus the low, subtracted by the low-calorie foods, and whether this amount of activation corresponded with the fasting insulin levels of each of the different subjects. So for those of you who don't know much about fasting insulin is the amount of insulin in your body when you're not eating, and when you have subjects that have high amounts of fasting insulin, it's a suggestion that they may be pre-diabetic. So high fasting insulin also therefore suggests that you might have subjects that have high activation with high calorie foods might have high amounts of fasting insulin. Again, this is another region that we know is part of the reward pathways. This is the insula. And this also gave us similar slope here with fasting insulin versus the amount of activation. We can also look at another measurement other than fasting insulin. This one's called insulin resistance. So when you are in a disease state or when you're diabetic, your amount of insulin sensitivity decreases, and therefore the negative slope. So when you have high amounts of activation with high-calorie foods, it shows that those subjects tend to have lower amounts of insulin sensitivity. Again, this is in the anterior cingulate, also related to the rewards pathway, and we saw a similar trend too. Next, we looked at what we really, really wanted to look at, and this is quite difficult, which is whether areas of that activated more in high-calorie versus low-calorie also saw this trend, and we did see this also in insula. And when correlated with insulin resistance or insulin sensitivity, those areas that have higher activation with high-calorie versus low-calorie foods, those subjects also tended to have low levels of insulin resistance. Similarly, this is a region in the putamen insula, and we also saw this trend. So after I did the fMRI work, I went back to focus on some of the work that our lab was an expert in, which was Alzheimer's disease. And Alzheimer's disease was very interesting because it has a very distinct disease pattern. More importantly than that, right now there are no in vivo methods of imaging Alzheimer's disease. Currently, the gold standard is post-mortem pathology. And that creates a lot of problems. There's no real standardized way of monitoring disease when the subject is still alive. So what does it look like? So here I've shown a sort of a diagram that, from the Alzheimer's Disease Association that kind of shows what the disease pattern of AD looks like. And it starts, goes anterior and posterior, and it loops around, eventually loops around. And this pattern is very distinctive and occurs in every AD patient. And we wanted to see if we could detect a similar pattern with the MRI methods that we're developing. So this is a chart sort of showing what the current methods are. So if you can see here, this is normal, abnormal. So normal and abnormal based on the method that we're using. Here's preclinical stage of AD or dementia. Here's MCI, which is mildly cognitively impaired, and here's full-blown dementia. So the further left on the curve you are, the earlier you're able to detect the disease progression. So right now, the standard, the earliest detection method in vivo is measurement of amyloid beta accumulation. And beta amyloid is basically the protein that they found that is most associated with Alzheimer's disease. And they form plaques that disrupt neuron signal transmission and all kinds of other problems. And right now, the way to measure this is you can either tag it with a radioisotope and do PET imaging, or you can measure the amount of beta amyloid in the CSF. And unfortunately, both these tools are not really useful as screening tools because, firstly, PET, you have to inject yourself with radioactive isotopes, which is not ideal, and you don't want to do that very often. With CSF, you are doing what they call a lumbar puncture, or otherwise known as a spinal tap, unfortunately, which is not a fun procedure to do. With MR, currently structural MR is over there, so we really want to move it further this way. And I believe that with diffusion MR, when we finally get to the level that we believe we can push it to the max, it would be somewhere around here. And that would be great. We would be comparable to what PET and CSF can do. So why diffusion MR? Well, in Alzheimer's disease, you see these plaques that form amongst the healthy cells. For example, here are Alzheimer's cells. These are healthy cells here. And these cells tend to disrupt the diffusion signal. And so plaques, beta amyloid plaques. And with diffusion MRI, we're able to measure the neuronal structure in the brain. And the hypothesis is that these plaques would disrupt the diffusion signal, allowing us to see differences between normal MCI and AD in their diffusion MRI measurements. So why diffusion? And what is diffusion? Well, diffusion is characterized by Brownian motion. Some of you may remember this from your early on physics classes and thermodynamics classes. Brownian motion is basically random motion due to thermal energy of particles, and it's freely moving when you're in a solution, for example, in the CSF. But once you have axons, they're structured to the brain, so now your diffusion is restricted to the space between the axons. And what we do in diffusion MRI is to measure this amount of restriction that you have when you're the amount of restrictions the particles have when they're diffusing in the brain. So we can characterize it in one simple way. One is that diffusion is isotropic, so it's uniform in all directions, or anisotropic. That means it's restricted along the axon, the direction of the axon. And we fit this with a tensor, hence the diffusion tensor imaging. And this tensor allows us to compute metrics that describe the amount of anisotropy, the amount of isotropy in that particular voxel. So one measurement is FA, which FA is normalized between 0 and 1. This would be 0. A perfectly sharp anisotropic ellipsoid would be 1. We also use measures like MD, which is mean diffusivity, and that's just the size of the ellipsoid, so lambda 1 plus lambda 2 plus lambda 3 divided by 3. The other cool thing about diffusion tensor imaging is that once you have this ellipsoid, you're able to find the primary eigenvector direction. So when you find this primary eigenvector direction, you know that this is where you can kind of in the axon you can kind of infer that the axon is moving in this direction. And the cool thing is, once you mimic the whole brain, you will have this vector field of where the estimates of where the axons are going. And once you have this estimate and this vector field, you can do this thing called tractography, which is basically moving along the vector field and reconstruct where you believe the neuron tracks are. So can any of you tell me which brain is a young brain and which brain would be an older person's brain? Any ideas?
Young to the left.
Young to the left? Young is to the right? Well, unfortunately, I actually found out that I actually made a very big boo-boo here. The resolution is slightly different, it's been pointed out. But actually, this is the Asian one, and this is a young person. And it's because of mainly most MRI people know this because we look at the enlarged ventricles. And that really means that there's a lot of CSF that has accumulated in the tissue in the brain. And that causes a huge problem with diffusion MRI. And here's another picture of it. This is the pathology when you take a postmortem pathology of the human brain. And why is this a problem? Because you have CSF in the brain, so you have this isotropic diffusion profile that you measure with diffusion MRI. And you mix that with your axons that are also existent in the same voxel measurement. And these are ellipsoids that have shape because of the fibers. And what you get is this combination of two, which is this ellipsoid that's a kind of intermediate between the two of these. And when you do FA measurements, you will have a pretty low FA for this, a higher FA for this. But then when you're trying to estimate, you're really trying to image what the neurons look like, but you have this extra CSF signal, and you have this measurement that's going to be lower than what you actually want it to be. And oftentimes what happens when we do this is that when we do statistical analysis, we find that these voxels are significant because it seems like they have decreased FA, but actually it's just this increase in CSF partial volume. So what do? We do a correction factor. And this is an image of the brain with tractography in it. The green is the hippocampus, which is an area we know is highly affected in Alzheimer's disease. And the blue is the corrected tracks. The red is the uncorrected tracks. And the uncorrected tracks has this crossing that we know does not exist. Moreover, it doesn't recover the fornix tract, which is indicated here in blue, which we know exists. Furthermore, when we did simulations of this, when we started mixing single fiber and CSF components artificially created, we found that when we do the tensor estimation, we saw that the error angle increased exponentially as we increased the CSF fraction. We also found that the shape, the FA value, also decreased significantly as we increase CSF fraction, which makes sense because you're mixing the neuron with the round signal, and as you increase the round signal, the shape becomes more and more isotropic. So what that means is, what we think is happening is that because of decreased FA values, when you do tractography ends when you hit a region that you know is no longer in the neuronal tract. And we know that because there's a certain FA threshold that we stop at. The other part is, can't see this very well, but this is an enlargement of this region, and I show the vector field with and without the correction. We found that when we don't correct for it, the vector fields are actually wrong. And when we do tractography, we have two thresholds. One's an FA threshold, the other one's an angular threshold. That means we don't expect tracks to loop back upon themselves. So at a certain angle, we'll terminate the tractography. And we also believe that that might be part of why there's so few tracks that are reconstructed when we don't do the CSF correction factor. So in summary, it corrects not only for the low amount of FA, but also corrects for the direction of the primary eigenvector.
Can I ask you a question about this? So one of the theories about Alzheimer's, especially in women, is a loss of white matter, a loss of myelin. And I would imagine that the loss of myelin would cause a change in the CSF.
Yes.
So if you correct, are you not correcting the thing you actually want to detect?
Yes, that's an excellent question, actually, and that's people have asked me many times about that. And the thing is, yes, when you decrease the amount of myelin, the amount of CSF does increase because there's space in between, so it fills up with CSF fluid. So that could be one scenario. But the problem is that the decrease is more significant than, well, I should put it this way. There's two things that could happen. It could be the tract that is disintegrating and CSF flooding in, and therefore decrease of FA values. Or it could be the tract is displaced because it's there's atrophy in the brain. And we believe when we look at this that the correction that we're doing is due to atrophy and not due to the disintegration of tracts. Partly because of the behavior. So there'll be a lot of tracts and then it hits a region and then suddenly it would just disappear. And that's not biologically possible. And therefore, and we also know from the anatomical imaging of the white matter that there's a lot of atrophy. Tracts do die away, but there's also a lot of warping and displacing of tracts. And we see this CSF correction factor, the weightings are highest when you're right around the ventricles. So we believe that what we're correcting for is actually when the tracts deform and CSF starts going in. Of course, there's no way of knowing for sure, but at the moment, I think, and I didn't show it here, but another study followed up on the work that I did and did it on a large scale with a lot of subjects. And they showed that in the large cohort that they were able to do FA measurements that were more correlated with clinical measures than with the correction than without. So they believe that this correction is actually doing something for the deformation and not the disintegration of myelin. So that kind of concludes the highlights of my previous work. I'm really excited to talk about two current really exciting projects that I'm working on right now. The first one is ICA multi-fiber tractography. So I've talked a little bit about tractography just now in Alzheimer's disease. One of the cool things you can do with tractography is to guide surgeries. And in surgeries, it's really important that you don't sort of damage key tracts because they might decrease the quality of life of your subjects significantly. And one of the tracts that's really important is the corticospinal tract. If you damage this tract, your patient tends to lose motor function, and therefore their quality of life becomes very low. So even though you want to be aggressive at removing tumor, you don't want to damage these tracts. But these tracts are very complicated. I'll show you a diagram later. And we really don't know, and with each person there's so much variation, you really don't know where they lie. So DTI has diffusion imaging has a potential of accurately delineating, reconstructing these tracts so that surgeons can know that they're not damaging them. The second part is some work that I've been doing in machine learning with some features that Natasha and her collaborators have created of the hippocampus. So they've surface rendered the hippocampus and looked at the deformations. And this is the work I talked about a little earlier. With the baseline MRI scan, we can predict what the neuropsych scores of the patients are for 48 months. And that's important because we then can know how to plan treatment for the subject. So first, DTI tractography of neurosurgery. So we're very excited this year. I submitted our work to the Mackay conference, and there's a tractography challenge, which is a group of 6 different methods people in diffusion tensor imaging, or diffusion MRI, and diffusion MRI tractography. And we're all trying to accurately reconstruct 3 cases for neurosurgery. And at the meeting, we're going to have a panel discussion, and we're going to have the neurosurgeons there. And they know what happened, they know where they cut, and they know how well the subject performed. And they have the MRIs to go with it. So we've, Nirhark and I have done some work on, and Daryl have done a lot of work at trying to reconstruct as best as we can with the methods that we have. And hopefully when we're on site, we'll be able to show off our work. And hopefully we didn't perform too badly relative to everybody else's. It's been accepted, so I hope that means that we didn't perform too badly. So why multi-fiber? So I've explained one problem, which is the increased amount of CSF, right? The other problem is fibers in the brain cross. So you might have a voxel in the brain that has one fiber going that way, another fiber going this way, and you're measuring the intersection point. And with the current tensor model, you really only assume that there's one fiber in that voxel. So for example, we have fibers going in the vertical direction. You have another set of fibers going that way. And when you do the measurement of this crossing, you really don't know where you can't do anything because you're assuming single fiber, but it's multi-fiber in this region. And we've seen this in our work. There's certain tracks that are harder to reconstruct without when we try to do with this assumption. Together with that, your FA measurements are wrong. So you have a high FA, which means a very intact track in one direction. You have also high FA in the other direction. Also know that this track is, in good shape. There's not much myelin degradation there. But when you put it together, you have this low FA because it's a fiber crossing, right? To make it worse, if you have a set of track that's that have degraded, so from lots of lines to one line, and you have a certain diffusion profile with that, and then you put it in a crossing region with another set of tracks going the other way, and you try to measure this, what do you get? You have 0.6 in your original measurement, or the measurement that you want, and then you have this contamination of the other track going across at 0.9, and what happens is you have this thing that's in between. So this degradation here from 4 tracks to 3 to say the 4 tracks is 0.9, it's decreased to 0.6, but you only measure the small change, whereas there's really a huge change in this fiber track. And this is a huge problem when we're trying to do measurements of where tracks are going and also measurements of how intact the tracks are. So what did we do to solve this problem? So Dr. Singh and Alec, who was a graduate student at USC in 2010, he graduated with this piece of work, independent component analysis of streamlined tracktography of the human brain. Basically, they use this mathematical technique called ICA to try to reconstruct tracks, multi-fiber tracks where there are voxels where there are crossings, and try to separate one track from the other. And why do we think that ICA is useful in this case? It's because ICA used to be used for voice separation, blind source separation. So we have 2 characters here. Some of you recognize who they are. Each of them is saying something. I won't tell you what they're saying, but they're saying something. And there are sounds coming out of their mouths, right? And it's picked up by 2 different microphones. So we have 2 sources and 2 microphones. And what we can do here is blind source separation. So we can, based on the statistical properties of their voices, we can try to separate their voices even though we don't know any other properties of them. But we have to provided we knew how many sources there are, and we know that there are 2 here. Similar with the brain, we can have one fiber that's going this way, another fiber that's going this way, and we can try to separate them. And we separate them by using, instead of microphones, we have measurements of these fiber crossing regions. So the sources are basically neighborhood voxels from the center voxel. So they have a crossing in the middle, but the MR actually picks up that crossing signal even in the neighborhood, the neighboring voxels. So why do we need this? So we take a look at the corticospinal tract. This is corticospinal tract. It's not a simple track. It's not just some fibers going straight. There are multiple crossings, especially up here. And sadly, a lot of tumors develop sort of in the cortical regions and near the fiber crossing regions. And so it's really, really important that we're able to resolve this fiber crossing. So what kind of improvements does our method allow? So here you can see in purple here's the tumor. Here are the tracks that we reconstructed, the corticospinal tracts. I labeled this single fiber because we assume that there are only single fibers in each measurement, in each voxel, so every measurement voxel that we have. Here's the multi-fiber ICA technique. That means we assume there's at least 2 fibers in each voxel. And I'll give you a zoom view. If you look at the zoom view, it's pretty dramatic. This angle is kind of hard to see, but with the single fiber, you see some penetration of the tracts with the tumor. But with the multi-fiber, you can see a lot more penetration. And what this would mean in a neurosurgical case is that if a surgeon saw this one and was like, oh, okay, we can cut off quite a bit of this without damaging the corticospinal tract. And in this case, it would be quite a different picture. There's a lot more penetration. He would try to probably avoid as much of this tissue here as possible. So here's one of the videos, beautiful videos that Daryl created. This is the reconstruction of the tract that we reconstructed versus the tumor up here. And these are pretty stunning images, and we hope that these videos will really help the neurosurgeons sort of guide their surgical planning. And they've given some of the methods people pretty good feedback in terms of what they would like to see in videos is definitely a great way to visualize it. Perfect.
May I ask
Thank
a
you.
question before you change subject?
Mm-hmm.
So you have two fibers crossing.
Mm-hmm.
So you're trying to determine the crossing locally, but the fiber is an elongated structure.
Mm-hmm.
It comes from somewhere where you can actually make a good measure. So you can measure 0.9 there, 0.6 there.
Mm-hmm.
Why can you not use some form of predictive coding or column filtering to determine how the fiber, so you measure elsewhere and use the assumption of the fiber to determine the property of the fiber in the region of crossing?
That's a great question and a great answer as well, because in fact nobody has done that, and I've proposed that a couple of times, trying to do that. And really they're looking at, I guess in imaging we tend to look at things voxel-wise. We look at one voxel in the surrounding region, but these structures are really elongated structures, and people tend to focus on one region instead of the whole brain. One of the things that I had proposed that I couldn't do this time was to do precisely that, to use the tractography to guide the image reconstruction, so that we know these structures that go in this direction. We can use that whole structure to guide our reconstruction. So I unfortunately don't have a good answer to that, whether that works or not, but it definitely is. I think a lot of people are going to come to that conclusion as well. It's definitely the direction of the field in the next couple of years.
I also have a question on the ICA technique. If you help us comment on it's using the blind source decomposition. I think it's premised on having
yes.
a lot of measurements relative to the number of sources.
Yes.
Is that accurate, first of all?
Yes.
And so can you bring it home now to your application? How is that satisfied in your application?
Yeah. So in our application, we have what we assume is that in the center voxel, that's the voxel in question right now. There's a crossing, and fibers are not going to be just in one voxel, so they also exist in neighborhood voxels. So if we assume that that's one measurement, that'll be the center voxel. Then we'll also have measurements in the neighborhood. So we actually have a configuration to do, I think it's 9 or 12 neighborhood voxels. And so we have those 9 measurements, and we assume that there's, at the moment, we can reconstruct only 2. So we set the amount of number of sources to 2, and then so it's about a 2 to 9 ratio in our problem. So we think there are 9 microphones and 2 sources.
In reality, however, you have approximately how many fibers do you expect in each voxel?
That's a huge debate. So at first, people were like, in the beginning, was saying that, oh, you know, the single tensor model works fine. There are beautiful images that come out of that. And in normal subjects, it's true. It works quite well considering how simple the model is. But with the Alzheimer's patients, it falls apart very quickly. We get these really weird things that don't look like anything that we would expect in the human brain. But now they're really trying to look at these smaller tracts, and I think a general accepted number now is, I think 80% of the brain has fiber crossings. And now they're determining whether it's 2 or 3 fiber crossings. So mathematically, the methods people unfortunately are saying it's 2 because 3 is very difficult to reconstruct. So we're wishful thinking. We're arguing that it's 2 because 3 would be very hard. personally, I think it could be 3. It could very well be 3. I mean, I've seen the pathology slides. These are really complex structures. The human brain's pretty amazing. And from just the sort of subjective idea of how the space is, it's very likely that there are crossings of 3. 4 is, I think, less likely, but 3, I think, there are probably a lot of. But the question is, what are we also, we know that there are limitations of the techniques, but the question is how far we can take it even with those limitations.
So you feel that you cannot support 3 with your measurements that you have?
We cannot. So the other end of this that I haven't talked about today is scan time and also the acquisitions that we do. So when we're fitting the ellipsoid, the advantages of our technique is we're using relatively low number of gradient directions. That means we're only taking samples, about 25 samples in the human sphere. And then there are guys at Harvard who do high resolution, and unfortunately it takes hours to do, so they put a macaque in the MR and they scan them for I think 36 to 48 hours. Then they get a very much better angular sampling of the voxels, and they can do much higher computational techniques. And we're quite excited about that. That code, the data hasn't been released yet, but we're all hoping that he'll release it at some point to all of us to play with. But then you run into computational issues when you have that many data points too. So, but we're hoping that if he's able to do it with that many number of samples, then we may be able to then judge what it looks like in the human brain and build techniques that are equivalent, so we know there's some gold standard and we're able to do that. And Marcus Reckley's group actually recently had a simpler method that was pretty interesting. He used a dictionary approach. So he has some data, I think, from Ben Wedin as well, and he has these highly reconstructed tracks. And basically what he does is he has a whole dictionary of single fiber crossing, double fiber crossing, triple fiber crossing, and he just creates this massive dictionary, and he just applies that to each voxel and tries to match them. And his results are surprisingly good, even though this is a very crude and mathematically, very greedy method. But I think with the computational tools that we have today, we haven't really explored. There's so many different methods. So last but not least, this project I'm really excited about because it's the first time that we're really able to predict what a subject looks like in the future, and it seems to be working pretty well. So how do we do this? So a lot of the work that we've done recently have all these statistical machine and classification techniques. It's really helpful to put it into a linear algebra framework. So I'm going to use this to sort of guide the methodology of my machine learning technique. So in the case of our machine learning techniques, we have a set of input features, and these features could be things like the sex of the subject, the genetic status of the subject. But in our case, we also use imaging. And sometimes we use voxels, and I'll talk a little bit more about that. But basically, we treat all of these inputs as features. So we can put it into a matrix. So here's a feature space. So these are, say, you can imagine it could be sex here, and then genetic status, and then voxel 1, voxel 2, whatever it may be. And then we have the number of subjects. So for each feature, we'll have n number of subjects along here. And in the general linear model framework in linear algebra for machine learning, you have a set of model parameters here. So basically, each one of these features is weighted by a number. And here I have time point t equals to 5 because I have 6 months, 12 months, 24 months, 38, 6 months, 48 months. And for each of the features, I'll assign a weight in calculating whatever my output predictions are. So it's a simple linear algebra operation. So we put in input features, we multiply by some model parameters, and we'll get some output predictions. In this case, our output predictions are 6, 12, 24, 48 months, the neuropsych score, so how how well mentally these subjects are able to do in a series of tasks, for example. So, seems pretty simple. The difficult part really is how do we solve for the model parameters? And this is where it gets interesting. Traditionally, you could just kind of really iterate it until you minimize the error, and then you have some output that you already have, so you try to solve that. We found out that that's firstly computationally very difficult. Secondly, sometimes computationally impossible. And also, you tend to do overfitting and other problems that you might have. We found it very useful to put a priori knowledge into the way that we fit this W matrix. And one of the great ways of doing this, especially in this temporal framework, is the way that the cognitive scores behave in time. So, for example, this is baseline. Say this person is performing pretty well in a memory task. At 6 months, they start decreasing. 12 months, it's decreased quite a bit. 24 months, it's recovered a little bit. 36 months, it's decreased again. 48 months, it's increased again. It's not very likely. We found that these cognitive scores and the people who design these cognitive scores want them to be very robust, and therefore they have very low standard deviations. And this is not an accurate representation of what real life is. Instead, we believe that these cognitive scores over 48 months should be relatively smooth. So what we did was we added a temporal smoothness penalty to our model estimation, and I'll show you the results later. It actually helped it quite a bit. Not only did it decrease the dimensionality of our problem, it helped it get the global maximum, global minimum, but also it improved our output predictions.
Can I ask you a question? So there are two beliefs I could imagine. No, just let them get up. The two beliefs I could imagine putting for this particular graph on the left. One is what you did, which is, well, it is really slow, and to use the smoothness plus the data to determine a curve. But the other, so if I look at the graph like the one on the left, after 12 months, you could imagine that there was a sudden decrease up to 12 months, and then the rest is steady state.
Yeah.
And so it wouldn't look like the graph on the right.
No.
It would look. So why do you think it's smooth?
I think so. Yeah. So this is. I think the other thing that I think even if it was like this in baseline, it is a smoothing. It's just how big your kernel is, right? So if your kernel is relatively small here, this would still be sort of a big drop here. And I think that's part of why this method works, is because actually when we do the training, we adjust the smoothing kernel and see what the output is. So for example, if my smoothing kernel here is smaller, then we would be able to take this drop a lot more rather than this gradual.
Okay.
And also the amount of weighting between the two.
So part of this smoothing is to avoid that oscillation, right? So you have to have. So in some respect, I think you have to have a smoothing that is as thick as that beam.
Yes.
So I'm not sure you can really do what you're saying.
Right.
I mean, if you make the smoothing too narrow, then you don't do anything. It's just going to look like the region. Of course, you could say, well, so smoothing is a model. You're simply stating a belief of what the process is, and you're saying, well, that's the best model for the particular data you observe. But you could say, well, in that case, I'll do a different model. I'll do set of an inverted error function or whatever. It's another model.
Right.
So I guess my question is, why that particular model? I know you say this, we believe.
Yeah.
But I mean, well, I'm giving you a different belief.
It's definitely possible that some of these tests may have. It has to do with the design of the cognitive scores, I think, and how the gradation is set up. And all these scores are normalized and designed in a certain way to behave in a certain way. So they actually collect there are lots of subtests, and they collect it over time, and they index it in a way to create certain behavior. So I think the reason why we did it this way was that the psychologist was telling us that this is what they designed the score to look like in time. So
psychologists.
I really don't know how it would actually behave, and also the answer I got also was that there's variations in depending on who's administering the test and what forms of test is used. So I suppose this is one way to do the estimation. Certainly you can fit other functions as well. I think this was also computationally the easiest way to do it as well. So, sorry. So that's one constraint that we have, the temporal constraint. The other thing that we noticed was that when we're looking at the model parameters for estimating any kind of biological system, they tend to be relatively sparse. And it logically makes sense if you say you're trying to predict how this person's going to behave in 12 months. You don't want a set of model parameters to say, oh yes, your sex matter is 0.1%, that one matter is 0.1%, it's even throughout all of them. That doesn't really make sense. There are probably some features that are more important than others. And so what we do is we impose, the simplest way is to impose this penalty called lasso penalty when you're doing the estimation. And that ensures that there are weights that are significant, and then there are some clearly zero weightings in some of the features. And that's a pretty common thing to do. But if you look at these weights, it also kind of doesn't make sense if you think about the temporal hypothesis that we had, right? So here you're saying that at 6 months, feature 1 is important. At 12 months, feature 1 is no longer important. But then at 24 months, feature 1 is important again. And at 36 months, feature 1 is important again. And then 48 months, it's not important. That makes no sense in terms of biological systems or in terms of the disease progression. So what some people have proposed is what they call temporal group lasso. So we say, okay, we still want the sparsity, but we want continuity in time. So once you select it, you have no choice. You select it throughout time. But then if we think about a little bit more, well, when we talk to people who do Alzheimer's disease research, they know that, if this person had a stroke, you know that in the 6, 12, 24-month time frame, they're going to have some serious problems. And that's going to give you a lot of information. But in the long term, it may not be that big of a deal. Maybe that's a bad example. But for example, other examples are like cholesterol and high blood pressure. It's not going to affect anything in the short term. But in the long term, it really could make a difference. So what we proposed was a few sparse group lasso. So in this case, we encode the smoothness that we talked about earlier, but also continuity in time. So we say that, for example, here feature 1 is important 6, 12, 24 months. And feature 2 is important later on for 36 and 48 months. And we believe that this really makes sense in terms of the disease model that we're trying to fit this to. Now we come to some features. So people have fed a lot of different information to the system, and we believe that if you feed it the right information, it will perform the way that it should. So one of the things that we found just in normal statistical analysis is that the hippocampus is very, very important in Alzheimer's disease. It's an important indicator of how well the person is doing. And more than just the hippocampus, the way that the hippocampus deforms as a person ages is a strong determinant of how well the. So what we proposed was this method of doing a surface rendering of the hippocampus. So you form a wireframe around it. And you look at, in this case, multivariate tensor-based morphometry, which basically means for each voxel in each of the subjects, how far has it moved away from the norm or the mean. So the further it's moved away from the original point, the higher it will be. And it's multivariate because we're doing this as X, Y, and Z. And when you're doing all three of them, it's quite noisy. But you really want the net amount of deformation. So instead of using all three, we decided to take the determinant of the Jacobian matrix, which is the vector of the deformation. And that gives us a much cleaner map. Still has the hotspots that you see, in the multivariate TBM. And we believe this is a better model of that. But then we also realized that there could be a lot of shifting up and down this way. And that may just be error in the way that we fit the wireframes together. And that may not be actual deformation that's of interest. So we found that another way of looking at this is we define a medial line. So for example, here that I've drawn in black. And we want to just look at displacement relative to the medial curve. And we found that, but this makes a lot more sense. It seems to make sense. So these are really the ways and the type of features that we fed into the system. And so what did we get? 6 months here, similar to the CAD system that I did earlier. We're trying to fit the 45-degree line. I've been doing that a lot in the last couple of years. So predictions here, the system's predictions. These are the actuals of the subjects. And you can see here, it does pretty well at 6 months. 12 months, still doing pretty well. 24, you can see some of these are starting to fall off here, not doing so great. 36, getting there, it's not so great. And 48, really, it's not doing so well. You also notice the number of dots, unfortunately, decrease. And that means some of the subjects of the cohort of the study that we have, unfortunately, has dropped out over the months, and we haven't been able to track them. But overall, I would say the system's working pretty well. So now we have these estimations. We are able to estimate these different time points and what the subjects are going to be like. The great part of this machine learning framework is you can look at the model parameters and really get information out of that. So how do we get those curves that I showed you? We got those, our cohort, and we divided into subgroups. And we use some of them for testing and some of them for fitting. But each time you fit it with a different sub-cohort, you will get a slightly different model parameter matrix. And so we're really interested in how these model parameters vary as we change the different input cohorts. And what we can do after we have all these is to calculate something called the selection probability, which is how likely is a certain feature to be selected at 6 months, 12 months, 24, 36, and 48 months. And we're hoping that this information will also be helpful to the people who study the biology of the disease progress. And we hope that we can correlate this subjectively with some of the observations that clinicians see. So this is really the power of this method, and this is the work that I have to do from now. This is the future work that I have. So this is really all I have today. Thank you very much. Here's an outline of the publications that I have. These are the ones that are for obesity and rewards pathway fMRI, some diffusion MRI and the Alzheimer's disease work, machine learning and MTBM, which is the newest work that I've done. And I did some work on diffusion MRI methodology, including that CSF correction, imaging informatics, and the work that I did previously. And finally, bone age here with ARC quite a while ago, but still work that I am very proud of. Finally, thank you to my community. For those of you who don't know them, we have Dr. Lepore, who is my advisor, Norberto, Dr. Morales, ARC, and Wolf. Thank you, guys, for being here today and spending time to critique my work. And of course, thank you all the other people who've been able to help me. Couldn't have done this without you guys. Firstly, Dr. Singh, of course, and Christian Nayak, who chaired my qualifying exam. He was very kind to do that. Dr. Leahy, Ming Law, and Helena Chu were my clinical advisors, and a lot of this work they've really advised me on, gave me a lot of the ideas, as well as the lab members that really have contributed to my work quite a bit. So that's it. Thank you. Open up to any questions you guys may have. Guys,
I'll start. I think there was a slide, the video that Daryl had of the tumor and the multi-tracks. He said it helps the surgeons in creating better plans for surgery removing the tumors. And do you have any sense of how much better that is in terms of like spatial recognition of those tracks and of the tumors, and then whether or not that's an actual functional difference in the removal process? And I guess I'm trying to get a sense of like size and accuracy of that.
Yeah, that's a great question, and sadly, what is in sort of the research and development realm and what is actually used in surgery is quite different. In actual surgery, they just really look at the slides of MRI and they try to plan it. And even the tumor, they don't really have these 3D renderings yet.
Sure. So
they're not even using 3D rendering. So this is kind of like a couple of steps ahead of what they're doing. And I think it really depends on the neurosurgeon as well, right? So I think I'm guessing that at this conference, the neurosurgeons who are coming are more used to doing this step. And then this is extra work for them, right, to analyze and have this extra data. And it's definitely not their standard of care right now. So I guess the short answer to that is no, I don't think we know how this will really affect the neurosurgeons. I think they have to embrace it first and willing to try it before we even know whether this impacts them. Theoretically, from just discussions with them, it seems like a powerful tool. But I think before it's widely accepted, it really has to be very polished, and we can be, all the different technical things that I can say. Oh, I'm not fitting the model properly and all that. They don't really want to hear that. So I think we really, as the methods people, have to clarify that and say we're very certain that this is true. This one may not be so certain. How certain are we of where the tracks are and where the tumors are and how we put it together? And I think also standardization across all the different methods, right? So each person might reconstruct it differently, might look different, might feel different depending on the lighting or the, and I think that really requires some standardization before we can really have an objective measurement of how well our tools are going to impact neurosurgeons.
Since you have done a lot of good work, of course. Some of the most intriguing results you present today have to do with the tractography results you have. You fairly pointed out a number of questions, not that we want to find many intelligent people for the years to come. My question, basically, I should list your comment. It's not that you give me an answer because I don't think anyone is able to do that at this point. But your comment with regard to tractography in general as an approach to understanding this phenomenon. And in particular, address the question of how much you think your results or your data are affected by the state of alertness of the brain of the particular individual. For instance, does it make sense to have a human subject perform a certain task if you are interested in a particular part of the brain to track, etc. Again, I would like to hear your comment on how much you think the result is affected by the specific task that the person is performing or the state of alertness in there.
Yeah, so I think in tractography, there are a lot of variations in terms of I think that the part that I haven't talked about is the acquisition of the images. And one of the problems in MRI is the standardization of methods. And with different vendors and different MRI systems, you get slightly different contrast. And I think that's something that we're trying to figure out, like how much variation can we tolerate? How much variation is acceptable, and how much variation is because of the system and not because, as you said, of the physiological state of the person. And we don't know. And in fact, actually, I spent 6 months working with Dr. Singh on this for a while. And we had a hypothesis that when there was visual stimulus, that the diffusion pattern might change. And that might be some of the variation we see. So we actually did, and Harka and Daryl and Bryce will remember this, we actually put somebody in the MRI and flashed really strong cube of light at them, and we tried to see if the diffusion signal changed. And it's a very low SNR sort of experiment, so we weren't able to find anything conclusive. But there's still a group within diffusion MRI that believes that when there's stimulus, that there is a change in the diffusion And that's important not only because that would be a great new functional method to look at the brain, but also that would mean that we have to look at our current data to make sure that we're not, measuring something that we're not intending.
Thank you.
Oh, yeah, just a quick question. To what extent do you think intensity and homogeneity could affect the analysis that you performed, especially if you can have a head coil and that compensates a lot? But is there anything left that still affects?
Yeah, so that's actually a very good question. So I talked a lot about FA, this measurement called band fraction anisotropy. And the reason why we keep on using it, even though a lot of people say, oh, this is not really a great measure for what you're trying to do, is because it's normalized. So you're measuring the shape of the ellipsoid. So whatever inhomogeneity, as long as it's consistent, will get canceled out, basically. So that solves the big problem with that. But really, it's a way of getting around the problem. So we're losing something and using that, be restricted to that FA. And I really think that there are some ways of correcting for these different inhomogeneities, but with MRI, because you're acquiring the frequency domain, you're not in the time domain. It's a mathematically very difficult to see when you do the Fourier transform, what happens. And in fact, here, Dr. Haldar, who's a new professor at WE, is working on that problem. He doesn't believe in using the Fourier transform because of that specific reason. So, and I think that is a big problem for MRI that doesn't exist with X-ray. X-ray, you can just kind of do denoising in space, and you don't have that complex time domain, spatial domain problem.
Do you feel that any of these techniques can be predictive of guidance in treatment in patients with Alzheimer's? Can we use that to see how certain procedures affect or do not affect what treatment has been being tested?
I think you bring up a very good point, Dr. Wolf. Right now, I think imaging is it's a pretty hot area, and a lot of people who do animal studies have started using them. And I think that's really where it is right now. If they are able to show consistently in animal models that they're able to look at treatment effects with the MR, then I think we can translate that back to humans. And I think with humans it's difficult for IRB and other reasons to be able to get really good traction in using and showing that. For more clinical trials, are just very expensive to do. As you know, YEHAB has one, and that's why we're trying to do that with the YEHAB study. He's actually treating them, and we're really trying to look at the treatment effect. But talking to Helena Chu, for example, she doesn't believe that, unfortunately, that there's going to be much funding to do that kind of work. Really, what we have to do is do animal models and try to look at that, and then go back to the NIH and say, maybe this could be applied to human models, because it's very expensive to do.
I just want to make a comment about a lot of the techniques that Sintra has worked on apply generally to many diseases, not just solely at Alzheimer's disease. And so, going to treatment, there are other disease states out there that have more clinical funding for in terms of medicinal treatments, like one being MS actually, and we're actually starting an MS drug trial that has a large imaging component in it, and probably some of the work that Sintra has done will be applied to that particular set of patients. So it's one of those things where it's specific to any branch. It can be widely distributed among various different studies.
Do you have a favorite project, and if so, how far you would like to take it from what was presented today?
That's a very difficult question because I only worked on one project with you, so.
Forget about Bonnet.
Okay, Bonnet aside, I think so. Hopefully, the future plan is to work on fetal imaging and neonatal imaging. And I've worked on very old people, and now it's kind of the young people. They're both the most challenging cohorts for medical imaging, and where they're also the biggest impact is we don't know very much about the development process, nor do we know much about the aging process. These are the two processes that are very interesting to scientists in general. So I think, yes, I think that my favorite, in short, I think the most rewarding work that I've done, I think, was with a lot of the machine learning work that I've done. And I think it's partly because I feel like it's applicable to so many different problems. And I think it's incredible how that set of mathematics translates again and again, and it comes up again and again, whether it's fuzzy logic or statistical analysis. That really, it fits all the frameworks, and it really is very elegant. And it works, which is really impressive. So I think that's definitely one of my favorites. Of course, modality-wise, I've kind of made a clear determination that MRI is my favorite now, and for many reasons, because you can acquire so many different contrasts in one scan. X-ray, you only get one type of image. MRI, you can get flow, you can get function, and that's really exciting.
When you go into fetal, what age before birth can you start?
I'm not sure. Did you know?
Earliest I have seen fetal being done is at 14 gestational weeks. That's really the most practical at which you can even detect a brain in the MR, but most often they start at 20 weeks because that's when you get a clearly visible fetal brain image.
It would be interesting to see how, from a temporal point of view, these fetal results then translate after birth into the neonates. And
I think we talked about that with Dr. Vanderbilt, yeah, a little bit about that.
Yeah.
So hopefully we're seeing, because a lot of children are, especially with neonates, they are, they're born early, so they tend to have certain problems later in life, and they're really interested in seeing if there are certain things that they can do to improve that. And surprisingly, there's not much data on that. So I think to definitely work on that. Thank you, everybody.
So now the committee needs to discuss it. We have to leave the room at that time.